All right, this is where we left off, cross-sectional view in different zones of the sarcomere. So if you cut in the I band, you only see the thin filaments. That's what you see in the far left there. That's what you're expected to know. The I band is the light band, but you have thin filaments. If you cut anywhere in the H zone, only thick filaments. The myosin filaments. If you cut right down the M line, M is middle. M line. You still have only thick filaments. But if you look at that picture, you can see that the M line also has, you can see the lateral proteins that make connections between the filaments to hold them in place. You see proteins that make lateral attachments to hold thick filaments in place. That's what I'm going to write. And then in the zone of overlap, you see thick and thin. That's the darkest band. Zone of overlap. So I'm just going to write thick and thin. They're both present. So in the model, you know, study those cross-sectional views and make sure you know what you're looking at. And they're labeled there. H zone, the M line, which also has the thick filaments with the lattice structure, thick and thin, and only thin. It's shown pretty clearly. It's not too hard to figure out once you figure out what's going on. And so in studying the sarcomere, there are things I haven't mentioned yet. So let's talk about my muscle fiber structure a little bit more besides the sarcomere. Here are terms I haven't used yet. So with these terms, study this picture here. Look at the whole muscle fiber and see how they pull out just a portion of it. So this is a portion of muscle fiber. We just looked at a sarcomere, so we're getting a little bigger here. And so we learned about these terms. Let's talk about the other things I haven't mentioned yet. Study the sarcolemma. That's the cell membrane. Okay, it's not the endomysium, it's the cell membrane. The thing about this cell membrane, it's not a flat sheet. There's going to be all of these invaginations where it kind of like burrows in. Well, let me just show you. So you have this flat cell membrane covering the cell, and then there's these deep invaginations right there every once in a while that traverse into the cell. And those are called T-tubules. The T stands for traverse, because they dig into the cell. And, so sarcolemma. So the triad is actually comprised of some of those T-tubules and what's called these terminal cisternae. Triad. Triad implies three structures. Okay, it has one T-tubule, and these two terminal cisternae. So terminal cisterna is a word that means end sac. Terminal means end. Cisterna means sacs. So you have two end sacs, and that's a triad. Okay, again, this is where calcium is released. Calcium is released from the end sacs. Okay, the T-tubule is going to receive an action potential. So there's a nerve impulse that can travel across the surface of the cell. But when it gets to these little invaginations, the T-tubules, the signal is going to go down. When the signal gets deep inside the muscle fiber, these end sacs, so maybe they're like, well, you can see how they're illustrated here on either side of the T-tubule. So end sac 1, end sac 1, T-tubule in the middle. So those three things is the triad. So what I want you to know is when the action potential travels down the T-tubule, there are calcium-sensitive channels that will open, because what I'm not telling you yet is these end sacs are filled with calcium, but they're trapped inside the sac. So here's my little calcium balls. the goal is to release the calcium from the end sacs for muscle contraction. But you only want to do that when you get a stimulus. That would be the nerve impulse, the signal that will stimulate some channels in here to open. And when you liberate calcium inside the cell, draw some thick and thin filaments here. Here's my thick filament with the M line, and then the thin filaments that overlap. The goal is for the calcium to interact with the contractile proteins, and I'll go into details of that later. For now, I'm just trying to teach you that the T-tubule with the end sacs is responsible for releasing the calcium when there's a nerve impulse. That's what you see illustrated there. Now the end sacs are just a part of this entire blue thing. The entire blue thing is called the Sarcoplasmic Reticulum, and we abbreviate it SR. So let me write that on the board. SR, Sarcoplasmic Reticulum, filled with calcium. The end sac is just the ends of this particular structure. Okay. The end sac is the end of it. That's where you concentrate the calcium until when it's released, it'll permeate the contractile muscle cells there. And I think that's all the detail I see. I think it's important to remember that when we studied the sarcomere, we studied a flat picture. But remember, these are kind of cylindrically shaped. Okay. And what they also show you here, there's a lot of mitochondria present because you need ATP for the muscle contraction. So note the mitochondria. Many mitochondria. Well, let's remember, you need ATP. And we'll see that hydrolyzed later. Well, anyways, if you look at the different models, you should be able to recognize these different things I just gave you notes on. So for example, on the picture top left, what do you think this blue stuff is? T-tubule. The beige stuff is the sarcoplasmic reticulum, but if you just look at the end sacs with the T-tubule is a triad. So on here, if I kind of like have you identify these three things, I'm going for the triad. But just the orange is the T-tubule, and the white structures that kind of are adjacent to either side are the end sacs, filled with calcium. I like this one because they also show the axon terminal, the nerve ending, which will stimulate the muscle in the first place. Okay. All right, so that's that model. Let's. Here's a picture from your book. And they show the nerve impulse going down the T-tubule, and that stimulates these channels to open for the calcium release. And they show the calcium kind of permeating the sarcomere structure. So again, the triad is located at the A-I band junction. Let me write that down here somewhere. Here's a histological picture of the triad. The red arrow is pointing to it. Triad. So here's light, dark. At the junction of light, dark, that's where A meets I. see A band and I band there. And you can see the triad right there. So that's an important detail to know. If you want to look at the outside of a cell and you strip away the endomysium, which is covering it, there's the endomysium, there's the sarcolemma. You can see the little perforations, which are T-tubules, so they're not really perforating the cell. They're just kind of diving deep, because think about it, this structure has muscle fibers all the way through it. Let me go back to this picture here. The T-tubule system allows the nerve impulse to travel to even the innermost myofibrils. Because if it were only on the surface, you wouldn't be able to stimulate the myofibrils on the inside. So the T-tubule allows the nerve impulse to spread throughout the transverse, all the way to the inside of the muscle fiber. Okay, back to where I was. Now I want to make a comparison here. We've been talking about skeletal muscle, but in smooth muscle, they have a more rudimentary design there. So this is for skeletal muscle. With the T-tubule. It's, in my opinion, it's better developed, okay? Whereas smooth muscle. In smooth muscle, you have a single cell with a solitary nucleus. In a skeletal muscle fiber, it's multinucleated. There's more an elongated cylinder shape. So let me draw like mini nuclei in there. It's multinucleated. If you look closely, it has these T-tubules. On the smooth muscle, if you look closely, it doesn't have T-tubules. It has these kind of like slight indentations, and they call them caveoli, instead of a full-on T-tubule, but it still works enough so you can get smooth muscle contraction. It has a rudimentary shallow invagination called caveoli. Not the T-tubule. But let's get back to the skeletal muscle and talk about the myofibril, because I want you to understand the details of actin and myosin before we proceed here. So if you look at one thick filament, it's actually composed of about 300 of those little myosin molecules. Thick filament composed of about 300 myosin molecules. So they draw one out there for you. Basically, there's a tail and a flexible hinge region so that the head can move. Okay. You got tail structure. In this flexible head region. And they have these binding sites. For different things. So the tail is like, this kind of looks like string cheese, right? But tail is like one little string. But that neck is flexible for movement. Flexible hinge is what they call it. Basically, it pivots. It allows the head to pivot. Allows myosin head. Sometimes they call them heavy heads. Heavy head or myosin head, same thing. Allows myosin head to pivot. That's very important for muscle contraction, that pivot of the head. So that's the head, the heavy head. There's a region that will bind the actin. Specifically, there's a little molecule on the actin called troponin, and we'll get to it. Binds troponin on the actin. But remember, ATP is required for living cells and for muscle to get that head to pivot properly and then detach and attach and pivot again, call it cross-bridge cycling. ATP's got to bind right here. That's for ATP binding. So when you break that terminal phosphate, it provides the energy for the work, the mechanical work of muscle. So that's why those details are important. So again, the heavy head, the hinge. Now if you look at the molecule it interacts with, the thin filaments, what we see is that the thin filaments, it's like you took two pearl necklaces and you intertwined them. That's kind of what it looks like to me. Now if you look at each individual, I guess, pearl, these little blue things, those are the actin subunits, these globular proteins. It's all protein here. So thin filament. Two strings, I use strings loosely, of actin proteins. They're the pearls of your pearl necklace, and they're kind of intertwined, wrapped together. But if you look at one actin subunit, you can see the active site where the myosin head will bind. I'm going to draw it like this. The actin subunit, that's the active site. The myosin head will bind to it. Oh, the other thing's there. Then there's this little rope cord that blocks the active site. Okay. Continue over here. It's a tropomyosin. Blocks active sites, during when the muscles relax, when it's resting. I'll say during rest. So if you have like the string of pearls, with the active sites, which I'm drawing as the little inner circle, actin, not to be confused with active site. The little plates. they're blocked when you don't want the muscle to be contracting with the tropomyosin. So let me just draw that as a little cord, literally covering the active site. My little black cord is the tropomyosin. You block the active sites during rest. Now if you look at the picture, do you see little yellow things? Look for yellow. Do you see it? That's very important too. Yeah, yellow. That's called the troponin. And that actually has 3 subunits right there. Maybe I'll use red. I'll draw it as like a red structure there. So troponin has 3 subunits. Okay. One is to bind calcium. One is attached to the tropomyosin, so it stays there. One binds to tropomyosin. What's the third one for? A brain fart. Let me look it up later. But this is the important one. Remember I said how, let's see here, calcium's important for muscle contraction? Because, yeah, that's really important. And it binds to tropomyosin. So don't confuse troponin and tropomyosin is the point here. I think the third one might bind ATP. I have to check. The point is, in rest, you want them covered. In a contraction, you want to move that tropomyosin off the active sites. So let's see if we have a picture of that in the next slide. Here's rest. These active sites, I call them binding sites, same difference. Covered, uncovered. They're like little olives, pitted olives or something. What color are the calcium ions in this figure? Red. You guys see little red balls? Red balls? Do you see how there's no red balls bound here? You hear there's red balls? Do you see the difference? When the troponin or the tropomyosin binds the calcium, it changes the conformation of the protein in a way where it sweeps the tropomyosin off the active sites that are exposed. So that's what I want you to see. When troponin binds calcium, it moves off the active sites, exposing them. So when they're uncovered, the myosin head can attach to the active site. Myosin head will bind active site. So that's the physical connection between thick and thin filaments. So they call that cross bridge formation, quote unquote. Cross finish that. Was there any questions on this slider or anything from just prior to that? There's a lot of terms there. What I have for lab, I have a couple of labs. One is for the bone tissue models I pulled out. And I just made my own key for them, and I just kind of printed those out. I'll put them by the model. But then I had a page to turn in. And if you're not sure what page to turn in, I just made copies, and it says, turn in this page. So that makes it pretty obvious, right? But then, see those muscle fiber models over there? I took pictures of those, and I made my key for it, for one, because there's two of them over there. I just made them for both. And then for the muscle fiber lab, oh yeah, just fill in the blank. So I know you guys get jittery with fill in the blank because it's like you can get it wrong. You fill it in wrong. So I won't, these two labs, I won't collect it until Monday, but I would like you to start working on it today. Because I know you guys get real like, oh geez, is this the right answer? And, took some of your lab time. So start on both of these today. I'll leave everything out here if you didn't make printouts because they're on Canvas. I'll collect both of them on Monday. Oh, you did? Yeah. You need more? I'll put the keys by the model.
She actually stopped. I thought they were joking around, you know, there are chairs, but bother me. He just likes the table. We should actually do that here. Is she getting it? I think she is. She wants
Foi lá? Ela tão uma luz. Niszkę i...
I don't know if they're right, but
That's Yuki, просто правильно. Я за 18 минут дал это.
Що ви такі? What is the exon
Oh, yeah, yeah. Yeah, that's the axon two over there. Here's your book. Does anyone have books? I'll show you the books. You can It's neuromuscular junction. Yeah, it's called the neuromuscular junction. I just wanted to show you the books you can look at it later. Yeah, I didn't cover that. So right here, if you look at this page, that junction right there is called the neuromuscular junction. Yeah, this is more detailed. I'll cover that column.
Wait, so tropomyosin is what?
That's correct.
M line. The middle. M line.
Okay, it's just the zone.
Yeah, so it's like two lines and the zone, and then the one with the line is the M line. And the Z lines are formal cells. Yeah.